Hi guys, welcome to 602 biology video in the genetics chapter, our first genetics chapter. And I already wrote in a couple of definitions. So if you want to pause the video and then come back. Okay, now that you've got those copied down, the law of independent assortment is review. We talked about that in the last chapter when we did meiosis and mitosis. And we said in meiosis 1, when the chromosomes line up in tetrads, it's random. So there is not any pattern to who gets to be on the right and who gets to be on the left, because when they separate, then that determines what gene goes to what gamete. And it's like dealing cards. You could get any one of the cards. So that indicates a degree of randomness, which gives us some variety. We have a new one. We've got the law of segregation. And what that says is during gamete formation, the alleles separate, giving gametes one copy of each gene. So we did that in meiosis. We saw that. We didn't talk about this law necessarily. Usually save this for this chapter because it really does come into play when we start to do these guys. These are Punnett squares. And you'll notice our title is a monohybrid cross. Monohybrid cross means we're looking at the inheritance of one trait. The inheritance of one trait. And so what we're going to do is we're going to look at how we use these Punnett squares to determine what we can expect in the offspring. So what we're going to do is we're going to consider the top part of our Punnett squares to be mom's portion. And let me see, what color do I want to use here? I'm going to kind of pick a light color. So we put an allele, and since we're only doing one trait, we'll have mom's alleles will go here and here, and let's just keep it pink and blue. Pink for mom and blue for dad. Dad's alleles will go here and here. So, and when I say here and here, I mean in this square and in this square, and mom's will be over here. So as we go through this, we're going to be using letters. And after a while, you quit thinking of them as being genes. And you just start thinking of them as being letters. But you always have to remember is that, oh, my pen's done. Rats. Yeah, that's done. Let's see. There we go. We're always talking about alleles. These are what you could possibly inherit. So let's take, for example, blue eyes, where capital B is brown and lowercase b is recessive. And Mendel figured this out with pea plants. So let's say I have, let's say mom is heterozygous. And let's say dad is heterozygous. Well, what does that mean? So we learned in the last video that if you're heterozygous, it means that you have one copy of the dominant and one copy of the recessive. So we put mom's alleles here. Mom has a brown gene, a brown allele, and she has a blue allele. Dad has the same. What we're going to do is to represent fertilization. So when we bring the letters down and across, that's the equivalent of fertilization. So we say that when we bring the letters down and across, that represents fertilization, where we're creating new life, we're forming a new human. So what I'm going to do is I'm going to bring this capital B down and this capital B down. This means that it was a sperm that had this gene and an egg that had this gene. Because remember, when we form gametes, gametes are haploid. They only have one copy. So if it was an egg that had the dominant copy and a sperm that had a dominant copy, the offspring would look like that. And that's the next important thing. These boxes are potential offspring. So this is an important point. And the other important point is that the boxes represent potential offspring. Okay, so let's keep going. Now I'm going to bring the lowercase b down and the capital B over. And you always, by convention, always want to write the capital first. So this is going to be capital B, lowercase b. I bring this down, capital lowercase. And over here I have lowercase, lowercase. And I'm going to pull my screen down a little bit, pull my paper, so we can have some more space. So here's what you need to know. This does not mean if you have brown eyes and you're heterozygous and you marry a man who has heterozygous brown eyes, that if you have 4 children, 3 of them will have brown eyes and 1 of them will have blue. This is a probability, a probability. So the Punnett square provides probable genotypes and phenotypes. Probable, and the key word here, and I'm going to underline this in red, is probable. So if you look, I have 4 possibilities. I've got 1, 2, 3, 4. Possibility number 1 is homozygous dominant. So this means that I have a 1 in 4 chance of being homozygous dominant. I have a 1 in 1, 2, 3, 4 chance. So I have the same chance of being homozygous recessive. But what about heterozygous? 1, 2. I've got a 2 in 4 chance, which would simplify down to 1/2 of being heterozygous. For review, let's go over your fractions. So on a monohybrid cross, which means we only have 1 trait, you could be 1/4, you could be 2 out of 4, which equals to 1/2, or you could be 3 out of 4. So do you remember from math class what percents we have here? This is 25%. If you put this in your calculator and you divide 1 by 4, you get 25%. 1 divided by 2 is 50%, and 3 divided by 4 is 75%. And those are the most common percentages that you're going to see when we do a monohybrid cross. Again, let me emphasize something. If we had, let's say we're doing frogs, and I had a bucket of frog sperm and a bucket of frog eggs, and I kept throwing together a sperm and an egg for a trait. Let's say this was for a type of skin or something different than color, eye color. That doesn't mean, maybe I would get, if I had 10 frogs, maybe so I should get, let's say frogs have blue eyes, or, we would have, if we had parents and they had 10 kids. What you would expect is you would expect 2 of them to have blue eyes and 8 of them to have brown, roughly. But maybe we get 6 that have brown and 4 that have blue. We don't always match the probabilities. It's kind of like when you flip a coin, you've got a 50% chance of getting heads. But even though you have a 50% chance, every time you flip a coin, you might end up with tails 3 or 4 times in a row. So that's probability. All right, let me give you a sample problem and see how you do. And I'm going to pause the video and write this out. Okay, I didn't have these preloaded, so I wanted to take a second and write this in. So this page is a very busy page. Apologize for that. So here's our problem. We're going to look at plants, and tall is dominant to short. So I have a capital T for tall, and I have a lowercase t for short. If you want to, you can pause the video, or just let me explain it, and then you can pause the video and try to work the problem out. So what would it look like if we crossed a short plant with a heterozygous plant? So in other words, what are the possible genotypes and phenotypes? I'm just going to put GEN, and in what ratio? So what are the possible ratios here of genotypes to phenotypes? And I abbreviated genotype and phenotype. So if you want to, pause the video and work this out and see how you do. Okay, welcome back. So if we are a short plant, that means we have to be homozygous recessive. And with plants, we really don't have a mom or dad, so it doesn't matter where we put them. And if we are heterozygous, that means that we have both a tall gene and a short gene. So when I do the cross, I'm going to carry this capital T down and bring the lowercase t over. I'm going to bring the two lowercase t's down. Got capital, lowercase, lower, lower. So when I look, here are my genotypes. I can be heterozygous, or I can be homozygous recessive. And heterozygous is my phenotypes. I would be tall, or I could be short. And if we look, my homozygous recessive is 2 out of 4. So that is 1/2. So this is going to be 50% short. I have 2 out of 4 that are heterozygous. So I'm going to have 50% that are tall and heterozygous. All right, now you're going to do some practice problems for homework. So hopefully, as you get into this, this is something you enjoy doing. All right, thanks for watching.